Dr. Melior. It is my pleasure today to introduce Dr. Tian. As you probably all know, Dr. Xia has received his MD degree from Harvard Medical School and then subsequently completed his PhD degree in biophysics from the leader of the universities in Asia and one of the leading universities in the world, actually Peking University. He then followed that by a postdoctoral fellowship here at UCLA in the Department of Medicine, and then eventually joined the California Nanosystem Institute, or CNSI as we know it, where he's now Associate Professor of Medicine. We're really excited to hear Dr. Xia talk about his work because he's a leader in the development and application of high throughput technologies to assess the bioactivity and ultimately the toxicity and the safety of nanomaterials. And I'm sure Dr. Xia will cover that, but as we know, nanomaterials are extremely prevalent in our world. They're now found in a plethora of products in healthcare and electronics, in food, clothing, cosmetics, you name it, and it's there. And yet, comparatively speaking, we know relatively little about their really interesting physical chemical properties and therefore their toxicity properties as well. So that kind of work is extremely needed. And I think as a testament to that, is the extremely high level of productivity and interest from in Dr. Xia's work, exemplified by his high number of articles, about 100 different articles that have been published through his research that have been cited over 50,000 times, which is really, really exceptional. I don't know if any of you really look into the H-index and the I-indexes, which are all these metrics of productivity, but Dr. Xia is definitely up there. And if you also look at the journals that Dr. Xia has published in, you'll find pretty well-known journals like Science, for example, as well as more specialized journals like Small or ACS Nano, etc. So we're really excited to hear about this really fundamental work from Dr. Xia today. Dr. Xia, the floor is yours.
Thank you, Patrick, for your introduction. So I'm honored to give this talk. So it's also my sort of a career journey of studying the cell death. So I'll give you some introduction on that. So all roads do lead to Rome. You know, people who have time have figured out that, indeed, from every village or, cities in Europe, you can go to Rome, by driving. So it's a metaphor for what we have found in cell death. So before I started my, PhD, there are known to have two mechanisms of cell death. One is better known, necrosis, that a cell under injury will die. And then the cell suicide. At that time, early 2000 or early 1990s, people try to figure out, some cell death pathways are controlled, are programmed. So that brings a hot research area you call the apoptosis. So these two different, cell death mechanisms are very, profound. That necrosis sometimes, often, the cell volume increases. On the other hand, apoptosis, the cell shrinks and the cell break up, the nuclear break up into the apoptotic bodies. So these now are better known for apoptosis. The pathways, the signaling pathways, the molecules, organelles involved has been figured out, like mitochondria, they trigger cytochrome C release, then apoptosis is activated, the caspases 3, 7 are activated and degrade the DNA's proteins, so the cell break up. So these are very well known right now. And at the cusp of this discovery, and 2002, the Nobel Prize was awarded to the 3 gentlemen here for studying, figure out the program cell death. By studying the organism called C. elegans. So Patrick will know this better if you have any questions to ask. So apoptosis seems very well known now, and people found out the other side of this cell death mechanism, necrosis. People found really the granularity in those cell death mechanisms. They found, necroptosis, pyroptosis, these, really weird names to say even necrosis originally has been thought to be a random, when cell under injury, assault by the toxicants, they die just, break up by swelling, these mechanisms. Now people found that they really actually are also under control, just like the apoptosis has been found out. And even for apoptosis, there are more granularity has been found out. Like it used to be considered, immune prevention, that the cell are disappearing silently. Right now, people see in the cancer studies, say some cell deaths in cancer cells are immunogenic. So by expressing the calreticulin, those eat me signals, on the surface that can generate the immune response. So as you can see in this slide, that there are so many, different cell death mechanisms are continuing being, and the new mechanism are being discovered, like mitosis, pyroptosis. It's hard to remember, but these are the current situation that people are finding more and more pathways cell death, just like the roads, although they go different railroads, but they all lead to Rome. The eventual destination is cell death. I just gave you some examples. Like necroptosis is probably the first controlled or programmed cell death necrosis pathway has been discovered, triggered by the TNF-alpha, and the binding to the TNF-alpha receptor then trigger a series of response leading to this pore formation by this MLKL protein. Then the cell swells and undergoes cell death. The pyroptosis is triggered by bacteria and the virus. And the recent findings showed that the SARS-2, the COVID-19 virus, can also trigger the pyroptosis cell death in the macrophages. So similar to the necroptosis, they also induce a pore on the surface of the plasma membrane. But at this time, the protein is different. It's called the gasdermin D. And the N-terminal of this protease, cleavage product will go to the plasma membrane and form a pore, and the cells undergo cell death. Then there are also more weird, cell death mechanism. For example, this called a neutrophil can undergo, this kind of mitosis cell death mechanism, that the nuclear membrane breaks down and the cell just expel all the DNA out to form the traps. So because the DNAs are, sticky, so the bacteria can bind to these nets. So that's a way of how can limit the infection. Also, these proteins can also trigger immune responses. So on the right, this video shows the neutrophils spelling out all the DNAs. So they can be stained by the PI to give out this red fluorescence, as we all know. And then another weird example of cell death is apoptosis. You can see in this video that the 3 cells actually, I don't know which eat one, another. So they finally merged into one. And the cells has been merged, can undergo cell death by the degradation process. And so why? There are like all the roads that lead to Rome, then there are so many different ways cells can die. So we all know that this is for the survival of the larger, organism. That's actually a defense mechanism for the body to go against, those toxicants, viruses, bacteria, infections. So the whole, organism can survive. So what does this mean for the nanomaterials? That's one of my main research area. On the nanoparticles. Nanoparticles are different from the chemicals, the toxicants we typically know, because they are larger, from 1 to 1,000 nanometers in size, and they have different compositions. They have different physical chemical properties that can give new functions to the materials. As Patrick introduced at the beginning, that these materials are increasingly used in our daily life, in daily products you are seeing. And so we study these using a high throughput screening method. So we incubate the nanoparticles with the macrophages. And from this heat map, you can see that there are different colors. The red means the cell is undergoing, cell viability loss or the cell death. And the green means the materials are relatively non-toxic. So you can see the nanoparticles can be divided into the toxic ones and the non-toxic ones. And for the toxic ones, what are the cell mechanisms? That's what I try to figure out. So some of the nanomaterials, like I show here, vanadium oxide, silver nanoparticles, copper oxide, and zinc oxide, they induce this apoptosis. As we know that the cell shrinks, so they round it up, and then later on they can break up into the apoptotic bodies. So these are well known, for most people, cell death mechanism, and the mechanism involved in this triggering this type of cell death is the involving oxidative stress, as we all know. We commonly know that the toxicants kill the cells by oxidative stress pathway and apoptosis pathway, involving mitochondria and inflammatory cytokine secretion. So I won't talk too much about this because these are relatively better known mechanisms. Then we found that there are some cell death mechanisms that are different from the apoptosis. It's a type of necrosis called the pyroptosis. You can see dramatically different cellular morphological changes. On this video that you can see, the cells, they swells, in an explosive way, like a bomb has been detonated in the cell. And on the right side, you can see the rare earth, one example of the rare earth nanoparticles, the yttrium oxide. They induce this type of cell death. The cell bubbles up and like an explosion has happened to it. So what's the mechanism involved? That we know the macrophage is allowed to eat up the particles, to clear up the particles by usually phagocytosis pathway. Then the cells can also take up the particles by other mechanisms, like a receptor-mediated endocytosis, pinocytosis. So after these, the cells take up the nanoparticles. They transfer these particles like they transfer the food, when they take up the nutrients from the surrounding of the cells, they transfer this eventually into endosomes, then early endosomes, late endosomes, then finally merge with the lysosomes. And along this way, the pH drops. At the lysosomal compartment, you can see a pH at 4 to 5. So there are many enzymes that are functioning at this low pH. However, to a nanoparticle, they are often, non-digestible. They are never seen by the cells, so they may generate adverse effects. For this one, I showed you that the rare earths can induce this pyroptosis, and when we incubate the rare earth oxides with a low pH environment, mimicking the lysosomal compartment, we see a very interesting phenomenon that the particle change from the spheres into this sort of a sea urchin-like structures. That happens really fast. That is what happens when the cells are taking up these nanoparticles. And what happens is once they are located into lysosomes, the particle transformation, right, from changing their shape, actually strip the phosphates from the lysosomal membrane. So the lysosomal membrane has been damaged, then leads to this caspase B release. What I stained the cell with a caspase receptor, a substrate that gave this red dots that are punctate form in the undamaged cell. However, in this rare earth treated in the middle, in the red box, you can see the caspase staining is lost. That means the caspase are released and distributed in the cytosol. And this leads to the activation of the NAP3 inflammasome formation, which are composed of the NAP3 protein, the ASC, the adapter protein, then caspase 1. This is a large protein complex that once they are activated, they trigger this processing of pro-I1 beta, a cytokine, major cytokine that are responsible for when you have an infection, you have fever, that the I1 beta is once the cytokine trigger this fever response in your body. And you can see that the rare earths not only damage the lysosome, leading to caspase B release, they trigger the NAP3 inflammasome activation to produce a large amount of L1 beta on the left side. And what happens, I won't go to too much detail, that we figured out the whole pathways of how the rare earth is, leading to the pyroptotic cell death, that they go in, they transform in the lysosome due to the low pH. They trigger NAP3 inflammatory activation, then caspase 1 activation that can cleave the pro-L1 beta into mature L1 beta get released. Then they also trigger the cleavage of the gasdermin D, as I mentioned before, into the N-terminal gasdermin D that they translocate to the plasma membrane to form the pore. Then the cell undergo, swelling, and the cell bursts up to die. At the lower right corner, you can see. So the difference between what I showed you, that some particle-induced apoptosis, some particle-induced pyroptosis, like the mechanism involved are the different caspases. These are the proteases, are activated in the cells that trigger the cell death pathways, like apoptosis and pyroptosis. And the major difference is the apoptosis is triggered by the caspase 3 and 7-6 activation. The pyroptosis, on the other hand, is caspase 1 and caspase 11. What I focus here is the caspase 1, that they cleave the pro-L1 beta into L1 beta, and they cleave the gasdermin D, the GSDMD protein that form a pore on the membrane to induce cell death. So one examples are the rare earths, as I showed you before. So, however, deeper exploration into the mechanism, we found that some of the particles, for example, in the red box, the silica nanoparticles, they can induce both caspase 1 and the caspase 3 and 7 in a single cell. So that's unknown before. So we want to figure out what exactly happens. Both caspases are activated. However, the cell chooses to die only through one way, not the other. So what's the decision making in this type of cell death? And you can see that the caspase 1 activation induced by the silica also induce L1 beta production. That's confirming, caspase 1 is activated. And what's the mechanism of cell death induced by silica? What we found is that different from the rare earth or other, particles, the silica has a different shape and sizes. They form this chain-like receptor, chain-like structure that they are not taken up by the macrophages. They stick, they all attach to the plasma membrane of the cell without being taken up. And over there, you can see on the right side, they induce the intracellular potassium loss that you can see that the silica nanoparticle induce a drop in this potassium fluorescent dye, staining PBFI. And they induce the loss of the potassium. And the potassium loss is actually another trigger of NAP3 inflammation activation. And you can see the caspase 1 are activated, then produce the L1 beta. And what's more interesting is that although they are not taken up like the rare earths, they also induce pyroptosis. This time their mechanism involves the membrane perturbation of the plasma membrane is sort of compromised, although not undergoing cell death yet. That causes the potassium efflux from the membrane and triggers the NAP3 inflammatory activation, then caspase 1 activation, producing the mature IL-1 beta and cleave the GSDMD. So they travel to the plasma membrane, form a pore. Then all the water coming in the cell, swell up. And this process is really fast. It caught like on the right, lower right corner, you see the cell literally blow up. And on the upper right corner, you can see that clearly the bubbles formed by these cells. So that's the silica-induced mechanism. Then we found another particle. It's called vanadium oxide. They trigger the caspase 3-7 activation. However, they do not trigger the caspase 1 activation at 5 hours, the earlier time points. However, although they don't have this caspase 1 activation, similar to silica, as I showed you before, they generate IL-1 beta. So that means the caspase 1 has to be activated. But here we show there are no caspase 1 activation by vanadium. Then at 5 hours. So we then decide to see whether, if we prolong the incubation time, whether we'll see caspase 1 activation. Oh, indeed. We see at the 16 hours, you can see vanadium trigger the caspase 1 activation. But at the 5 hours, the earlier time point, they don't. So that's why the vanadium can trigger the IL-1 beta production. However, what's their mechanism of triggering the NAP3 inflammation? So detailed study, we found that the vanadium actually dissolves in the cell culture medium, and the dissolved vanadium ion actually is an inhibitor of the plasma membrane sodium potassium ATPase that maintains the gradients, inside and outside of the sodium potassium that you all know. And this inhibition of the sodium potassium ATPase actually causes the potassium release from the cells because on the cell membrane, there are also some constitutive potassium channels that are constantly leaking potassium outside. When you don't have the sodium potassium ATPase to pump in the potassium, then you cause a potassium loss in the cell. So what you see here, similar to what I showed you before, the intracellular potassium are lost by the vanadium incubation. And similarly, vanadium ion can also trigger its loss. And the potassium efflux, or the loss of potassium in the cell, is a trigger for NAP3 inflammatory activation. That's why the vanadium, is producing a lot of IL-1 beta. However, the caspase 1 activation is happening at a lower time point. The caspase 3 is actually activated at the early time point. They do their job to kill the cell by apoptosis pathway. But at the same time, they also produce pro-IL-1 beta because caspase 1 is activated later at a later time point. So on the left upper right corner, you see the cell undergo the apoptosis instead of the sort of explosion or swelling induced by the silica nanoparticles. So the differences in the cell death mechanism involves temporal differences in the caspase activation. So for silicon nanoparticles, they are not taken up by the cell. They induce membrane perturbation, then potassium efflux. This happens fast. So the caspase 1 cleaves the gas thermin D into this terminal, N-terminal form will transport to the plasma membrane, cause this pyroptosis. At the same time, silica can also induce caspase 3 activation. However, the caspase 1 induced cell death involve less, complicated, mechanisms. You just need to cleave the gas thermin D, then they transport to the cell membrane to induce cell death. So that's easier and simpler. It goes faster. Then the vanadium also, it's a different story, that they induce this potassium loss at a slow pace. At the same time, they induce mitochondrial ROS generation, so they activate caspase 3-7 at an early time point. That induce apoptosis on the right side. Interestingly, caspase 3 and 7 can also cleave the gas thermin D that play a role in the left side of the pyroptosis. However, after the caspase 3 and 7 cleavage, these fragments of gas thermin D, they lost their function to go to the plasma membrane to induce the pore formation and induce pyroptosis. So that sort of also inhibited the happening of the pyroptosis pathway. So you can see that the cells are determining where to go, how to die. But they have all the components, all the tools ready in front of them. They activated, both caspase 1 and caspase 3. Now they have a choice, to see which cell death pathway I go. So that's, highly fascinating to me. So over the years, we figure out, not only the rare earths, the metal oxides that I mentioned before. We also studied the cellulose, the fiber structures, the 2D nanomaterials, molybdenum disulfide. So they all induce, different pathways. Sometimes they induce, caspase 3 and caspase 1 are activated at the same time, but these materials choose to die, only one way, either apoptosis or, pyroptosis. So that's what we found out, it's the nanomaterials really are, depending what type of materials they determine their particle cell interactions, that you either stay on the cell membrane or, you're taken up inside the lysosome damage, oxidative stress. So these are complicated, but, over the years, we figured out their, cell death mechanism. So that's this is the end of my talk. So I want to, quote Mark Twain's message here. I think if we change the man into the cell, it really summarizes what I talk about today. A cell who lives fully is prepared to die at any time. And in fact, the cells are prepared, to die, at multiple pathways, has the potential to go multiple pathways. So to undergo cell death. So that's the end of my talk. Thank you very much for your attention. I'm happy to answer some questions if you have. Thank you.
Thank you, Dr. Xia. And yes,